Okay, hello, everybody. Welcome to the second session. I'm Warren Wagner. I'm a research botanist and Pacific Island curator here. I work on other families too. But I have the pleasure to introduce the next two speakers, including the second of two Emilys. Dr. Emily Meineke received her PhD from the Department of Entomology in North Carolina State, and she followed up her PhD with a postdoc at Harvard and did a lot of work in the herbarium there. She is currently on the faculty of the University of California, Davis in the Department of Entomology and Nematology as an entomologist. She's especially interested in human activities and how they affect plant and animal interactions, especially in crops, crop wild relatives, urban trees, and so on, as well as plants to support rare insects. She is focused on utilizing information from the vast amounts of information that we've been hearing about so far today that are stored in the research collections of plants and animals to try and understand global change. She also heads up a art science fusion program at UC Davis, where she works with students and others from the community every year to create large public art displays depicting local ecology. And today she will speak to us on herbivory through the ages, applying paleoecological methods to herbaria to investigate modern global change.
Hello, everyone. I'm so excited to be here with you today. And thank you for the first two speakers for the wonderful introductions to herbaria and botanic gardens. I'm not sure if I'm going to be able to get this. Oh, yep. Okay. So I am someone who was trained classically as an entomologist, and I didn't really expect for my career to take a turn into herbarium collections. And so what I want to talk to you about today is how that turn happened and some of the things that I've done since herbarium specimens have become a core part of my research program. So what I and my lab study the most actually are whatever is flying around me, insect herbivores. So insect herbivores have been eating plants for something like 400 million years. And the diffuse coevolution between them and plants is thought to have resulted in most of the terrestrial biodiversity on Earth. So despite being small and mostly invisible, they have profound roles in ecosystems. So insect herbivores make up the bottom of food webs. They also aid in decomposition and on and on and on. So it's also true that insect herbivores can be profoundly damaging, especially in contexts where we want to grow plants and see them thrive. So for instance, even little amounts of herbivory that you might see when you're walking your dog, say a hole in a plant, they can have outsized impacts on that plant. So one example is that these birch sawflies, by removing something like 10% of leaf tissue from growing saplings, they reduce vertical growth of those trees by 30%. According to the UN, invasive insects alone cost farmers approximately $70 billion per year. So as I mentioned, our lab pursues research questions related to how insect herbivores and their relationships with plants change due to climate change and other factors that are changing with human development. And this is a difficult thing to predict. It's also a really interesting thing because insects and their host plants are super responsive to climate and environment. So on the one hand, this seems like it should be a really easy thing to predict. So what you're looking at here is a standard thermal response curve of an insect. Every insect behaves this way. So as temperature increases, metrics of performance such as survival, egg size, egg number go up until you reach a thermal optimum, at which point the performance rapidly declines. So given that mean temperature acts in this way on insects, we should be able to figure out where insects are living on this curve and then how climate change is going to affect them. However, of course, we're also experiencing extreme temperatures both in winter and in summer. In summer, maximum temperatures might kill off insects if they get too high. And in the winter, insects actually require chilling, like a lot of plants do, to complete their life cycle. So many insects diapause. So warming in that life stage alone could lead to the decline of insects. So really complex predictions about what climate change will do to them. It's also true that a lot more things other than climate are changing right now. So we have the introduction of novel compounds such as pesticides. We also have land use change. And one thing that is popping up as a really important driver of changing insect populations is the loss of host plants. And remember that as I go on. And that loss of host plants is primarily thought to be due to agricultural and urban development. So what we do know about insects right now and the cumulative effects of human actions on the environment is that across a lot of data sets, even though we have very little data sets actually on change over time in insect populations, the one that we do have show that there is something that people are calling the insect apocalypse happening, where we do see declines in insect abundance and biomass. And this is referring to a New York Times article that was written in 2018 about this topic. So there was a study that came out shortly after this New York Times magazine article that summarized that between 1976 and 2012, we lost a little less than 1% of all insect biomass per year, which is what you're seeing in those brown bars for terrestrial biomass. So 2 questions that motivate my research that have come out of these recent studies of insect decline. So given that insects are in decline, has herbivory changed over the past century? So herbivory is a measure of how insects are interacting with plants, how they are affecting plants. And we actually don't really have a lot of good data sets tracking this over time, which I'll show you in a second. And are insects declining uniformly? And so the data that we have that actually span the period of recent global change, they are typically abundance or biomass data. So we are limited in the data that we have about insect diversity. So if we actually look at the first statement that I made in number format, so what I did was I plotted study duration and study count for all studies in ecology that address herbivory, and they usually last between 1 and 2 years, which is certainly not long enough to assess the effects of climate change. And if we're to go back to this first slide that represents insect biodiversity, insect herbivore biodiversity, none of these taxa have been monitored over time such that we can actually determine how individual species are responding to global change. The only really good data that we have on insect diversity over time are for butterflies. So I went on a search for data for these 2 questions that I was really interested in building my career on. And what I found is that herbarium specimens are the most promising resource for being able to reconstruct these historical interactions over time. So it turns out that insects leave behind characteristics marks when they feed that are related to their mouthpart morphology. And so just looking at each of these insect groups, I won't get into detail, but they are all changing plants in ways that we can see and we can actually measure on herbarium specimens. And just in case you don't believe me, this is the oldest specimen at Carnegie Museum of Natural History, and it displays these holes which are probably signs of beetle feeding. A lot of the herbarium specimens that I have seen presented here today also have herbivory on them, and I will bet you that some of Dr. Premac's slides that he's going to show will also have herbivory on them because it is really ubiquitous on these plants. And so not only are there sort of generalized types of feeding like chewing that show up, but there are also types of feeding that we can track back to the insects that made them. So there is some hope of using herbarium specimens to track the diversity of individual species or clades. So what you're looking at here is a characteristic sort of half moon shaped piece of a leaf that's removed by a megachilid bee. And what you're looking at here is actually a type of damage called a leaf mine, where a tiny little moth or some other insect lays their egg within the leaf tissue, and then the larva chews and eats its way through the leaf, sort of making the leaf into a cafeteria and a house. The shape of this leaf mine and the, forgive me, poop inside of it are characteristic enough for us to be able to track this mine back to this individual micromoth species you see here. So as you've heard today, herbaria are really extensive resources. They're also digitized in a high resolution way that makes it so we might be able to use them at a broad scale to understand how plant-insect interactions are changing as plants decline and as insects decline. And so when I got to Harvard for my postdoc, I wanted to ask this first question I posed to you, which is how has herbivory changed over the past century or more? And it turns out that Harvard has a really rich collection from the Northeast, which is something you would imagine. And the Northeastern U.S. has had a lot of warming in winter. And so I got to Harvard and said, okay, I'll focus on Northeastern U.S. collection, which is digitized. And I'll start with this prediction that herbivory in New England has increased over the past century with rising winter temperatures. So because winters are becoming milder, we might expect that insects are surviving the winter better. Alternatively, because we're seeing widespread insect declines, you might also imagine that that leads to reduced feeding. And I chose, after many conversations with curators in the museum, to focus on 4 species that have outsized impacts in their ecosystems. So they variously support rare insect herbivores or provide acorns, various other functions within their environments. So then I had to take these species and figure out how to quantify insect damage on them, which turns out to be non-trivial because they are collected in all kinds of different ways. And primarily these specimens were collected either for other types of science or leisure. And so I came up with this system, which is very low tech, but I was really excited about it at the time. So you basically put a grid over the specimen, you choose grid squares randomly that have leaf in them, and then you write down whether they have a type of herbivory in them or not. And in this study, I focused on chewing herbivory, primarily made by caterpillars and beetles, because it was by far the most abundant type of herbivory on all 4 species. So then I ran into sort of another hurdle, which was that these specimens display what is clearly damage that's made by insects out of the museum before the plant was collected, but they also display damage by dermestid beetles and tobacco beetles and things like that inside the herbarium. And so I spent like 1 to 2 months or something like that of my postdoc just trying to figure out how to tell the difference between the two. And what I found is what also paleobotanists have found, which is that when live plants are eaten, they get what's called a reaction rim. So if you look at that top picture of damage, you can see that there's sort of a brown line around the damage, and that is the plant reacting to the insect herbivore. And then on the bottom right is a picture of damage that's made by tobacco beetles, which goes all the way through the leaf and in a very characteristic pattern. So once I was confident in my data from all of these species, I'm not going to trouble you with all the statistics and things, but basically we wanted to know how often these specimens displayed signs of damage. And we wanted to account for all of the different things that could be affecting the herbivory on these plants that had nothing to do with year or winter temperature, which were the primary predictors we wanted to test. So those are all listed there for you. And I'm just going to sort of jump into our results here. So the colors that you're seeing are different species. Along the horizontal axis is year that a specimen was collected. And then along the vertical axis is probability of herbivory. And the specimens are noted as dots. So if a specimen's on the top, it had some sign of herbivory in those 5 grid boxes. And if it didn't, it's on the bottom. So across these species, herbivory, the prevalence of it, increased by 23% from 1896 to 2008. And I'm going to show you another graph here that tests the second half of our hypothesis, which is that increasing winter temperatures have something to do with this. So across 3 species, we did find evidence that places and times that were warmer made it so that the plants were more likely to display herbivory. The one exception is Quercus bicolor, and I have an idea of what's going on here, but I'd love to hear if anybody else has ideas as well. So winter temperature is going to continue to increase in New England, and herbivory is likely to continue to increase as well as insect metabolic rates go up, their rates of feeding go up, and probably they continue to survive better. This might be a result of insect herbivore range expansion from south to north. It also could be higher abundance of native insects with warmer winters. So the focal plants were chosen for this study because they provide ecosystem services, and it would be really interesting to follow up and see how those are affected with this increasing herbivory. So what I really learned from this study, I think the most important thing, besides the fact that herbivory is rising in New England, and that seems actually kind of surprising given insect decline, is that herbarium specimens offer the opportunity to study plant-insect interactions more broadly in ways that we are otherwise just not able to. So I told you I would come back to the oak. So I think what might be happening there is the spongy moth, which is an invasive species, was introduced into the Northeast. And I think that maybe that rise that we're seeing over time in the Quercus in particular is due to that. So I think we have some combination of climate change and invasive species driving herbivory as it goes up. Okay, so before I displayed to you all of these diverse types of herbivory that are made by different insects. And so far, I've shown you the answer to the first question, which is the prevalence of common types of herbivory changing over time, which is chewing herbivory. But I really wanted to expand this question and start using herbarium specimens to address the broader question, which is, are insects declining uniformly? So we have all this abundance data for insects, we have biomass data, but we do not have good diversity data for the vast majority of insects. And it turns out that I am not the first person who has had the challenge of figuring out what's happening with insects while not having the insects available. So paleobotanists at this very institution have developed methods for reconstructing plant-insect relationships using plant fossils. And so what you're looking at here from A to H is different parts of plant tissues that have different morphologies of insect damage, all on pressed fossils. And in fact, this system has been used to track the diversity and prevalence of insect damage across warming in the fossil record. And so during the Paleocene-Eocene thermal maximum, a time when air temperature warmed 5 degrees in the span of 10,000 years, so it's thought to be as close of an analog as we can get to current climate change. What happened during that period is that the prevalence and diversity of insect damage also went up. So when I was studying herbarium specimens, I thought, why not apply this wonderful system to modern leaves in herbaria? And luckily for me, there was a damage type system already developed. And so Conrad Labandera developed this work in 1999 and then just continued to expand it through trainees. And this is one page of what is a very long damage guide that he has written for the damage type system, as it's called. And so each leaf is examined for these different types of damage in fossils. And the damage types are characterized by their specific pattern, shapes, and sizes. And of course, is only quantified if a reaction rim is present. And I tried to do this myself. I tried to get herbarium specimens and damage type them, figure out what kinds of damage were on there, and I should not be doing that. So I brought in a qualified paleobotanist, not just qualified, wonderful, Lauren Azevedo Schmidt, who has used this damage type system, used it very successfully during her PhD. And she is helping me answer this question, are insects declining uniformly? So at UC Davis, we had to choose a study species to focus on, and we chose valley oak. The reason we chose valley oak is it is a wonderful tree species it hosts something like more than 4,000 species of insects, birds, reptiles, etc. It serves as food or shelter for those species, and that's been documented in a project on iNaturalist. These are some of the species that it supports. And so at any given time, you can kind of go up to a valley oak or Quercus lobata and see all kinds of different interactions. So again, I won't get too deep into the weeds about this, but we selected herbarium specimens of Quercus lobata in California, where it's an endemic species, from 1901 to 2002. We made sure that those specimens covered time and space. And then we ended up with 70 specimens and something like 413 leaves that Lauren damage typed. These are some of the damage types that she found. So hole feeding on the top left, on the top right is a type of leaf mine. On the bottom left is another type of hole feeding, and on the bottom right is another type of leaf mine that's more serpentine, just to give you an idea of the kind of hard work that Lauren did here. Okay, so similar to the other stats that I showed you, in these models, damage type richness as a proxy for insect richness is one of our response variables we're interested in. We were also interested in seeing damage prevalence again because we have those data. And we have year as our predictor of interest. And then we also have 25 climate variables that we were interested in seeing, you know, how much they correlated with insect damage, because insects are so they are so responsive to climate. And one thing that I have not mentioned yet that seems really important is that Conrad and colleagues have also done a certain amount of work to show that in modern forests, damage type richness is also correlated with insect herbivore richness. And so this really is a proxy for insect herbivore diversity on plants. So I'm going to jump right into the results and show you a couple more graphs. So what we're first looking at is the effect of these variables on insect richness or damage type richness on the plants. So these different lines are different models, but they tell essentially the same story. So year, again, herbivory does seem to be going up over years in terms of richness on these oak trees, and this is not what we expected, and I will explain it. And then the other thing I want you to look at is mean warmest month temperature. So it also seems to be correlated with elevated herbivory rates, probably because a lot of insects are multivoltine and they go through more generations if they have warmer temperatures during the growing season. We also see with extreme minimum temperatures that as they go up, potentially insect diapause is interrupted and insects are not actually able to complete their life cycle, which is why when we are in times and places that have high minimum temperatures or more mild temperatures, that at least in California, these extreme temperatures actually lead to declines. So when we look at prevalence, it's much the same story, just with fewer significant variables. But again, temperature variability, so warm summers, cool winters are what the insects in California seem to be requiring to do their jobs. So let me just put all of this together in one clean package. So like in New England, herbivory is increasing over time in Valley Oak. Climate warming in summer is probably partially responsible, but so are other factors, because when we include climate variables with year in these models, year is still significant. So probably the introduction of non-native species in this system is important as well. Insect richness on Valley Oak is also increasing over time, which I will come back to. A little bit shocking. Some insects also seem to require winter chilling that fuels diapause. And so unlike in the Northeast, where winter temperatures that are milder are benefiting insects, it does seem like in California, at least the majority of insects do need that chilling. So here I'm going to give you a couple of important takeaways. Insect decline does not necessarily yield decreasing herbivory on all plants. It's the opposite in both California and in New England. And so how did these results fit with insect decline? Well, remember how I told you that a lot of insects seem to be declining because of habitat destruction that removes their host plants? The insects that are associated with valley oak and with the other plants that I showed you are associated with relatively abundant plants. And so what this suggests to me is that the plants that are sticking around, especially in abundance, are still supporting their insects. And so what we need to do, this lends credibility to the idea that a lot of insects are declining because of the loss of their host plants that the other Emily brought up to you earlier. Okay. I want to end with a couple of things. One is to thank all of you who continue to do this really hard work of curating museum specimens and collecting them without necessarily knowing how they're going to be used in the future. I have benefited profoundly from your work day in and day out, and I'm very grateful. And I'd also like to point to a project I worked on that's very different with artists using digitized specimens from Henry David Thoreau's collection to visualize plant decline. So these are his specimens, and the ones in cyanotype represent plant clades in decline in Thoreau's woods. And it wouldn't have been possible without natural history collections. And so I was also really excited to hear Lucia talk about making sure that collections are available to other groups of people as a means of showing their value and cultivating their value, because artists are really excited to use these resources and would be more if we were able to bring them to them and make them known. All right, this work required help from a lot of people, and thank you all so much for listening.
It's really interesting to see herbarium images being used for this kind of research. And my question was, do you have any idea the approximate image resolutions for herbarium specimens that would be necessary to best perform this kind of research? And perhaps that varies by insect group?
I am so glad that you brought this up. So I I'm such a proponent of digitization. The physical specimens are also invaluable doing this kind of work because you can get an idea of a search image better from a 3D thing than a 2D thing. So that's the first thing. The second thing is, I don't know it off the top of my head, but University of Connecticut's Herbarium has the most high resolution images, and it helped me so much that they had these really high resolution images. So if you go onto the Consortium of Northeastern Herbaria website and you look up Connecticut's collections, that resolution's great for
All
us.
right, thank you.
Hi, this is a fantastic talk. I was wondering if there's any way that herbarium specimens can reveal how natural enemies, such as parasitoid wasps and flies, are also changing over time in their prevalence on these plants.
Yeah, so I should say, I was hoping for an opportunity to say this, but my colleague Elsa Youngstedt is the first one who alerted me to herbarium specimens as a potential resource for this kind of work, because she counted armored scale insects on red maple trees. And they leave behind their waxy cover on the stems. And you can also see parasitoid exit holes. And so and you can flip over the scale insect covers and see developing larvae too. So it's not just the exit holes. And I've always wanted to go back and do that project with her of looking at parasitism rate. So I think there are lots of opportunities for insects that make different structures and also for looking at different trophic levels like that. Yep. Thanks, everybody.